is a fluid ejaculated from the male reproductive tract. Hence, it is a valuable tool to assess the reproductive health. In this video, I am going to tell you what is semen and how it is used to examine. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get their study material. Make sure you subscribe my channel. So today we are back with the next video on the reproductive system, that is about the semen and its examination. So first we will see what is semen, and then we will in the next part we will see what is semen examination. So semen is the fluid expelled from the urethral meatus at the end of the male sexual act or the male orgasm. So if you have seen the previous video on the male sexual act, you already know it's a reflex and it has three motor responses: erection, emission and ejaculation. So ejaculation is the last one and it is terminated by the ejaculation of the semen from the urethral meatus, which happens because of the contraction of many muscles, and it is a very forceful expulsion. So what does the semen consist of? It consists of sperm and seminal plasma. So now we already know that sperms are one which are produced by the testes, and the seminal plasma is the one which is produced by the accessory reproductive glands and the ducts, in which the major contributors, or you can see the major volume, is being contributed by the seminal vesicles and the prostate gland, and only small fraction of the volume is being contributed by the bulbourethral gland and the epididymis. So next we will see that how much volume of the semen is being contributed by the sperm and how much volume is being contributed by the seminal plasma, and in that also how much percentage of the volume is contributed by the major contributors and how much by the minor contributors. So you will see here that only 10% is being contributed by the testes in the form of the sperm, and rest 90% volume is being contributed by the accessory reproductive glands to form the seminal plasma. So for example, if you see that 2 ml is the ejaculate volume or the semen from the male you have collected, and now from that you can see that 10% of that volume of semen is only sperm, that is around 0.2 ml is being contributed by the sperm, and rest 1.8 ml is actually the seminal plasma. In that also 70% is by the seminal vesicle, and rest 20% is by the prostate, epididymis and bulbourethral gland. So in this 20% also the major contribution is by the prostate. So you can see that here that the major contribution to the volume of the ejaculate or the semen is actually by the seminal vesicle. So you can relate that when there is a low semen volume, you can actually relate you can first see that is there any problem with the seminal vesicle, because that is a major contributor of the seminal plasma or the semen. Now we will see what does the seminal plasma is having. So as I said that there are minor contributors, major contributors. So what does these contributors actually contribute to the seminal plasma? So first is the seminal vesicle, which is giving whatever it is giving, it is leading to the mucoid consistency of the semen. So in that there are many things which are contributed by the seminal vesicle, but I have highlighted the ones which are very important to remember. So we will see about those ones, like the seminal vesicle contribute the fructose, prostaglandins, fibrinogen and seminoglands. So you can remember these. Then prostate is giving a milky appearance to the semen. So in that you remember citric acid, cholesterol, phospholipids, fibrinolysin, zinc, acid phosphatase and prostate specific antigen. Then bulbourethral gland and mucus glands, they are majorly producing the mucus for the purpose of lubrication. Ions, calcium is very important. This is the one which is going to increase the mobility of the sperm. How it is going to increase the mobility of the sperm, we will see in the next video. And buffers to maintain the pH of the semen, since pH is very important for maintaining the sperm vitality as well as its mobility. And hyaluronidase, which is again very important. So we will see the function of them, broad functions of these highlighted ones in the next video, in the next slide. So seminal vesicle is contributing the fructose, which is giving the nutrition to the sperm. So if you remember the video on the sperm structure, we have talked about the metabolism of the fructose also, like that there are GLUT transporters which are present on the sperm membrane, through which the fructose enters inside the cell of the sperm. In the sperm there is mitochondria, so the mitochondria is going to metabolize the fructose to generate the ATP. Then prostaglandins are having the function, they are going to induce the contraction of the smooth muscles of the uterus and the fallopian tube, which will assist the sperm movement from the vagina where they are ejaculated to the site of the fertilization, that is the ampulla of the fallopian tube. Then seminoglands, they are secreted by the seminal vesicle, and they are going to induce a coagulation of the semen after ejaculation. So soon after ejaculation, what's happened, that seminoglands as well as a fibrinogen, what they do, they actually going to coagulate the semen which is being ejaculated. And this and the advantage of coagulating soon after the ejaculation is that it is going to hold that semen in that region of the vagina, in the deeper recesses of the vagina. So these two are important to induce the coagulation of the semen. So even if there is a collection of the semen for the purpose in the laboratory, in that also, if you see, first the sample will coagulate and then it will liquefy. Then there is a process called as a liquefaction which occurs. So fibrinogen is activated by the prostatic fluid enzyme. So some enzyme in the prostate secretion, they are going to activate the fibrinogen, and it will also coagulate the semen immediately after ejaculation, so that it can be holded in the deeper regions of vagina. Then we will see some important secretions of the prostate gland. Prostate specific antigen is the one which is going to, it is a protease which is going to liquefy the coagulated semen. So after ejaculation there is coagulation, after coagulation there is liquefaction which occurs after few minutes of the coagulation, and this is having a function. Apart from this, fibrinolysin and fibrinogenase, they are also going to dissolve that, they are also going to dissolve that coagulum which is found by the semen. And prostate specific antigen, very very important, it is an indicator of the prostate health. So sometimes if there is elevated levels of the PSA in the plasma, it can be an indication of the prostatitis, prostate tumor, or the carcinoma. Also, the prostate specific antigen is something which can hydrolyze the sperm motility inhibitor, which is seminoglands. So what happens when the sperm coagulates after the ejaculation? It is going to hold the sperms together. So as a result, the motility is very less. So when it liquefies, the motility increases. So you can say it is going to hydrolyze the seminoglands so that the motility could be increased. Then acid phosphatase, I can't find out the specific function of acid phosphatase, but it is an enzyme for the phosphocholine, which is actually a product released by the seminal vesicle. And then there is hyaluronidase. So this hyaluronidase is actually present inside the sperm, in the cytoplasm of the sperm, and it is being released into the seminal plasma, and it has a very important function of depolymerizing the hyaluronic acid which is surrounding the ovum. So that is going to allow its entry around the ovum and finally penetrate or fertilize the ovum. Then the next is about the semen examination. So now we know about what is semen, and we broadly know about the major components of the semen and also about the function of them. So now we can talk about the semen examination. So basically it is a kind of a laboratory test which is being done to assess the quality and the quantity of the semen. It has various applications. So the first thing is that it can be used to evaluate the male reproductive function. So basically, as I said in the beginning only, that it is the semen is the output of the male reproductive system. So it can be used to assess the health of the male reproductive system. So if there is any abnormality in the semen, that can be related with the specific gland. So if there is abnormality with the pH, we can relate it. Is there any abnormality with the seminal vesicle secretion or with the prostate secretion? Because the prostatic secretion is acidic, whereas seminal fluid secretion is alkaline. So their relative contribution is going to make the pH of the semen. So we can relate the things with a specific characteristic. Then fertility potential. So this is being extensively doing to assess the fertility status of a male. So like if there is, what is the percentage or total sperm count in the volume, in the ejaculated volume, as well as what is the percentage of the normal forms, like whether the sperm which is being released is having a normal morphology or not. So again, after knowing about the problem, we can choose the appropriate infertility treatment for the couple. And then efficacy of the male contraception. So for example, after vasectomy, what happens, what should ideally happen, that the semen after semen collection after vasectomy should not have sperms, or it has very little sperm. So that is a test which can be used to assess whether the vasectomy is efficiently done or not. Then how the sample is being collected, it is actually collected by self masturbation in the vicinity of the laboratory, near laboratory or at the laboratory, or at home in a sterile amber colored white moth and glass bottle after the lack of sexual activity, absence from the sexual activity for about 3 to 5 days. So then the semen examination includes basic examination, extended, and the advanced examination. So basic examination is the one which is regularly being done in all the laboratory setup. Extended and advanced are being done mainly for the research purposes. So the content of the semen examination I have taken from the WHO manual. So that manual is recently published in 2021, and all the content is being taken from that manual. So they have mentioned the semen examination has these three things. So I have added here. Now, what does the basic examination is having? What are the different parameters the basic examination is having? So the procedure for the collection of sample for the basic examination is this only. So first of all, after collection of the sample, the first thing to be done is the ejaculate volume. That what is the volume of the ejaculate? So normal volume is around ranging from 1.3 to 1.5 ml. So basically what happens that before collecting the sample, the weight of the container is being done. Then after collecting the sample, the weight is being done, and then there is a subtraction to get the weight of the sample. So now we know the weight of the sample, and we also know the density of the semen, which is 1 gram per ml. So from that the volume could be calculated. Okay, so the clinical significance of the ejaculate volume, since I said it's being the seminal plasma which is being making the major part of the volume of the semen, it is actually giving us a status of the secretory function of the glands and the ducts. Also it is being used for calculation of the sperm count and the non-sperm cells. Okay, then this point will be clear in the further slide. Then there could be instances in which the ejaculate volume is more than the normal limits. So that is called as an hyperspermia, which is seen in the case of the longer abstinence, inflammation of the seminal vesicle, because as you know, seminal vesicle is a major contributor. So in that case, if there is inflammation or certain kind of infection, it is going to affect the volume. Hyperspermia is a low semen volume, which is seen if there is an incomplete semen collection, poor abstinence, testosterone deficiency, retrograde ejaculation, seminal vesicle abnormality, hypogonadism, ejaculatory duct obstruction. Then aspermia is a condition where there is no semen at all. It could be psychological ejaculatory duct obstruction or retrograde ejaculation. So here one point to remember is that there could be that there are that these reasons are not going to be specific for a one type of abnormality. They are actually they are a kind of a journal problem. Okay, so if I can say that retrograde ejaculation, it could be responsible for the aspermia, as well it is a factor which could be seen even in the hyperspermia. Similarly, testosterone deficiency could be leading to hyperspermia, could be leading to the aspermia. So multiple factors could lead to the different conditions in the sample, in the semen sample. Okay, then Next factor is the color. So what is the color of the sample? It should be gray. So color is signifying something like if there is slightly yellow color, it could be a result of a longer abstinence. Yellow color in the case of the jaundice, and then there is red brown color if RBCs are present. Then as I said, liquefaction. So immediately after the ejaculation it coagulates and then liquefaction happens. So 15 to 30 minutes is the range at the room temperature it liquefies, and it is going to signify the function of the prostate specifically, or more specifically the prostate specific antigen. So depending upon the level of it and the function of the prostate, it can vary the duration of the liquefaction. Then ejaculate viscosity. So it should fall drop by drop. In case of abnormal viscosity, it is not falling drop by drop, it is actually falling as a thread 2 cm long. Then the specific gravity should be 1.028. So then the next factor is the pH. So for the pH, the procedure is the semen is well mixed and then it is spread on the pH strip, and then it will be left for some time, and then it can be compared it with the calibrated pH strip. It ranges from 7.35 to 7.5, and as I said that it is basically a relative contribution of acidic prostate and alkaline seminal secretion. So if there is increase in prostate gland secretion, there will be low pH. Okay. And if there is decrease in seminal secretions, again they will be low pH. Or in the case of the urine contamination also, they will be low pH. Vice versa happens in the case of the high pH. Then sperm concentration. Sperm concentration means how many sperms are present in 1 ml of the ejaculate. So basically it is being done by the dilution and the cytometry method. So in this video I am not talking in much detail about the procedure. If you want to know about the procedure, about the precautions and about the troubleshooting, you can refer to the WHO manual. I'm going to share the reference of it in the last of the video. At the last of this video, you can download from the Google, and you can refer to it. So normal range is around 15 to 18 million sperms per ml. And then from the sperm concentration, there will be calculation of the total sperm number. So as I said, ejaculate volume is used to calculate the sperm and the non-sperm cells. So you know the sperm concentration and you also know the ejaculate volume. So if you multiply these two, you will get the total sperm number, which ranges from 35 to 40 million per ejaculate. So there are two conditions which you should remember. Oligospermia is a condition where there is low sperm count. Okay, so it could be happen testosterone deficiency, hypergonadism. So again I'm saying there are numerous reasons which could lead to the oligospermia, so I am not able to, it's very difficult to list them all. And also these are the reasons which are general, which can be applied to many conditions. So it is not possible to give you the whole set of reasons which could lead to the oligospermia, but I have mentioned some of them to give you some clarity. Then there is azoospermia, like no sperm in the semen, which could happen after vasectomy. Okay, so next is the motility. So motility is a movement of the sperm. So sperm has to move in order to reach the ovum to fertilize it. So motility assessment is also very important. So you will see first about the grading of the sperm motility. So this is also given in the WHO manual. So rapidly progressive means it is covering a distance of more than or equal to 25 micrometer per second, and progressive means it is moving ahead in a forward direction, and also linearly or in a large circle, but it is moving forward. Slowly progressive means it is not having that much velocity, but it is moving actively either linearly or in a large circle. Non-progressive means there is active movement of the tail, but there is no progression. Immotile means there is no movement of the tail at all. So it can be, this motility could be assessed by microscopic techniques or by the computer aided techniques. So this is a percentage in which it is being given, like 40 to 43% of the spermatozoa, they should be, or the total spermatozoa, they should be motile. So in this what happens that we count 100 or 200 spermatozoa in total, and after which how many sperms are motile, how many sperms are showing the progressive motility, how many sperms are showing the non-progressive motility is being commented in terms of the percentage. So 40 to 43% should be the total motility, including both progressive and non-progressive. Then progressive should be 29 to 31%. Immotile could be 19 to 20%. Then there is a condition which is called as the Asthenospermia, which is absence of any motile sperm or a diminished motility. Then vitality. Vitality is basically how many sperms are alive, how many sperms are dead in the semen. So this is being done by staining the smear of the semen with the eosin nigrosin dye. So basically you can see here in this diagram, this is based on the principle of the integrity of the sperm cell membrane. So if the cell is dye, the plasma membrane is ruptured, so the dye could enter inside the cell and it could stain the cell. So if the sperm cell is stained with a dye, in that case it is a dead cell, whereas the cell, if it is not stained with dye, it is going to be live cell because its integrity of the plasma membrane it's still maintained. Okay, so 50 to 56% of the vitality is normal. And necrospermia is something which is the dead sperms. Okay, now this we have already covered. Now the last one, the normal forms, it is a normal morphology. So we know the sperm is having the head which is containing the acrosome, nucleus, then there is middle piece, then there is a tail. So the parts of the all the parts should be present and they should have the normal morphology. So how the morphology is being assessed is again by the smear formation and by fixing, staining, and then the microscopic evaluation. So 3.9 to 4%, again it is being commented in terms of the percentage, and you can see this is a normal morphology of the sperm. Okay, so tetratospermia is the condition in which there is abnormal morphology of the sperms, and globozoospermia or a round head defect is a kind of a defect whereas there is absence of the acrosome. So you can see in this particular slide that how many different types of the defects are actually present. So I have talked about this condition which is called as a round head defect or globozoospermia, in which there is no acrosome. So that head could be tapered, piriform, amorphous, vacuolated. They could be defect in the head, they could be defect in the neck and midpiece defects, they could be defect in the tail also, and they could be the excess residual cytoplasm. Okay, so this is a table which is showing all the normal morphology, the points of the normal morphology and the points of the abnormal morphology. This is also taken from the WHO manual. Then about the extended examination. So in the extended examination we talk about if the sperm is having the multiple morphological defects. Not only there is a problem with the head, but in the same sperm there is a defect in the middle piece also. That is called as a multiple sperm defects. Then sperm DNA fragmentation. There could be single strand or double strand breaks in the sperm DNA. Now there could be genetic defects of the sperm which is called as a sperm aneuploidy test, in which there could be decrease in the chromosome number, there could be increase in the chromosome number, trisomy or monosomy types of the defects. Then there could be presence of the WBCs in the semen, presence of interleukins, that is inflammation, and assessment of the immature germ cells, like there could be presence of the spermatogonia which should not be present in the semen. And then there could be the antibodies testing for the spermatozoa. So these are the extended examination. Okay, then biochemical assay also being done for the accessory gland function. Some of the you can see some of the markers or the secretions are the secretions of a particular gland. So like citric acid, zinc, glutamate transpeptidase, acid phosphatase are the secretions of the prostate. So if there is an increase or decrease in these levels, there could be a indicator that there is a problem with the prostate. Fructose and prostaglandin seminal vesicle, free L-carnitine, glyrophosphocholene, and neutral alpha-glucosidase. These are the markers of the epididymis. And also there could be assessment of sequence of ejaculation. So if you remember the video on previous video, we have talked about the sequence of ejaculation. The first to come out is actually the part of the ejaculation which is having highest number of the sperms and the prostatic secretions, followed by the vas deferens, followed by the secretions coming from the seminal vesicle. So in that, basically what is being done, that like there are you can say test tubes in a series, and in that test tubes, sequence wise there is ejaculation of the sample. So in that case there could be assessment of the sequence of the ejaculation. Then the advanced examination, such as like oxidative stress या reactive oxygen species testing, assessment of the acrosome function, assessment of the sperm chromatin, and also the channels which are present on the sperm, such as the Caspar calcium channel. So this is the journal report form, what is being mentioned in the WHO manual, that there is a patient name, that date of birth, and everything about that, what is the collection, complete collection, who yes or no, collection time, sample delivered, and if there is any kind of history of any infection, what is the volume, okay, and then all the, what is the visual appearance, viscosity, then liquefaction, then ejaculate, okay, whether there is any aggregation or reglutation which is being seen, then pH of the sperm, semen, sperm number, okay, total number, concentration, motility, vitality, morphology, non-sperm cells if they are present, accessory gland function also, what is the content of the fructose, what is the content of the zinc, what is the content of the alpha glucosidase, any additional comments. So this is about the whole manual, this is about the whole uh, the examination of the semen. So these are the references. This is the manual from where I have taken the semen examination. It is latest, published in 2021, and these are the books from which the content of the semen is being taken. So you can refer to these for more details. So if you like this video, please like, share and subscribe my channel, and till then keep learning.